Hold up. Before we get into this grant, here's what already happened. KD3010, a potential drug therapy, was tested in animal models for Huntington's disease. In this case, it proved it was safe and effective for Huntington's. KD3010 also has already been approved by the FDA for testing in human patients. Rock on. But isn't I AM ALS about ending ALS? Yes. And the good news is that Huntington's disease and ALS have shared pathways as neurodegenerative diseases. Neurodegenerative diseases means the cells in your brain or other nerves in your body, neurons, start acting out. For Huntington's disease, KD3010 has shown in animal models to prevent the cells from dying, neurodegeneration. It has shown it promotes lifespan extension, and it has shown it rescues function. Meaning, instead of losing function in, say, your hand, it is shown that it actually reverses that loss to regain function back. Reminder, if something is effective for Huntington's disease, it could mean it's effective for ALS. But no one has tested it in ALS. That's where we come in. So, I AM ALS is funding this grant to see if the compound will slow or stop the progression of ALS. Disclaimer, this will likely not cure ALS, but it could help people living with ALS. Here's what I AM ALS is funding as part of this $200,000 research grant. Number 1, Dr. Albert Laspada and his team at UCI Institute for Neurotherapeutics will insert the KD3010 compound into 2 animal models with an ALS mutation. One will have the ALS mutation TDP43, the other will have another ALS mutation, C9orf72, G4C2. Second, Dr. Laspada and his team are making neurons from stem cells of patients with ALS with the mutation TDP43. These will be treated in a petri dish with the KD3010 compound, and the different cell functions will be measured. If and only if the results are good, the next step is to try this KD3010 compound against ALS in humans.